Okay, this is the intro to our new unit, and the unit I'm calling Inheritance Unit. This will be Unit 6, and these are the two objectives that we're going to work on during the course of this unit. So by the end of the unit, you should be able to do these two things. Let's get this term here, inheritance, clear to start off with. So inheritance in terms of biology means how are traits, and by traits I mean characteristics. So it could be things like your hair color, your skin color, eye color, whether you've got straight or curly hair, whether or not you're lactose intolerant, whether, anything that's a characteristic that you have, whether it's a physical characteristic, something you can see, or a physiological characteristic, something that your cells do. Those are your traits. So inheritance is how traits are passed on. Okay, so how you inherit your characteristics. Now, looking at the first one, the first objective here, there's the verb predict, and this is my what happens if, or what do you get if verb. Because when you predict something, you're going to say what's going to happen. Well, since we're talking about inheritance, what this actually means is this. What do you get if, right? Little dots there. What do you get if? What do you get if a tall, brown-haired man has children with a short, blonde-haired woman? That's the kind of thing. What would the offspring, what would the children look like? That's what you're going to be able to do. And in order to be able to do that, we're going to have to do genetic combinations. Well, a genetic combination is what you might remember doing back in middle school. This is what we call genetic crosses, okay? And genetic crosses use something called Punnett squares. You may remember that term, okay? But that's one of the things you're going to be able to do by the end of this unit. And you have to determine the possible outcomes of various genetic crosses. Right, we've got monohybrid cross. We know that mono means one, okay? We have dihybrid crosses. We need to know di means two. So this says, what happens if a monohybrid crosses? What happens if a brown-haired man has children with a blonde-haired woman? I'm only looking at one characteristic. A dihybrid cross would obviously look at two characteristics. So if I've got a brown-haired tall man and a blonde-haired short woman, I've now got the hair color and the height. Those are two separate characteristics, and I'm going to predict, a kid's going to be tall with brown hair, or maybe they're going to be short with brown hair. Maybe they're going to be tall with blonde hair. Maybe they're going to be short with blonde hair. Then we're going to look at two characteristics, and then we're going to look at something called non-Mendelian inheritance. The non-Mendelian inheritance is stuff that doesn't really follow the rules. So, for example, any characteristic which is carried on the XY chromosomes, that what we call the sex chromosomes, the ones which determine whether you're male or female, any characteristics that they're inherited differently if you're a male than if you're a female, which is why, for example, you're much more likely to have a man who has red-green color blindness than a woman. It's the same thing if you look at, most of the examples we tend to look at are human examples, but any of you that have an orange cat, I almost certainly guarantee it's going to be a male cat, because orange fur color in cats is associated with the male cats 99% of the time. In fact, 1 in 100 orange cats will be female. Okay, those are kind of how we inherit characteristics that don't follow the set rules that were put down by this guy called Mendel. Right, he was a German monk who is actually referred to as the father of genetics, because he came up with the rules on and Punnett squares on how we do stuff. So, and when it says such as, that means these are examples, but there could be other things. So I might throw in 1 or 2 other examples of different ways we inherit characteristics, but that's what we're going to be able to do as a minimum. The next objective is kind of a cool one, actually. I think it's quite cool. It says recognize the significance of meiosis to sexual reproduction. Well, the first thing I want you to notice is that word is meiosis. It is not mitosis. We covered mitosis, prophase, metaphase, anaphase, telophase, when we did the cell cycle. Meiosis is, it's like a cousin to meiosis. But here's the thing. It does not say recognize all the stages of meiosis, right? We're not having to do the step by step by step by step. We've got to know the significance. And whenever you see that word, if something says, what is the significance of, it's basically asking you this question. Why is it important? Why is meiosis important? And trust me, it's very, very important, because what does meiosis do, right? Oh, let me get a pen that works. Good luck with that. Here we go. Okay, so meiosis has one job, right? And its job is to produce. It is the production of gametes. Now, you might know what gametes are. There are 2 types of cells in animals. There are somatic cells, which are cells that make up your body. Skin cells, nerve cells, bone cells, muscle cells, all the cells that make up your body. And then there's gametes. And gametes, right, you actually do know what gametes are, because gametes are sex cells. So we have the ovum, which is the proper term for an egg, the female sex cell. And then we have these little dudes here, which are called individually, they are spermatozoans. Spermatozoan or spermatozoa. You can see now why we only called them sperm for short. Those are obviously your sperm cells. Okay, so gametes are, female gamete is an ovum and the male gamete is a sperm cell. Okay, now that's in itself, that's important, because meiosis produces sex cells, and without sex cells there is no next generation, there is no reproduction. Okay, but here's the other thing that's key about meiosis. It helps, this is a great phrase, helps maintain genetic homeostasis. Okay, what on earth does that mean? Well, we know homeostasis is keeping things steady. Genetic homeostasis, if it weren't for meiosis, we would not be able to guarantee that every offspring has one complete set of DNA. Not a half set, not a double set, not a mixed up set. a complete set of DNA. All right. Well, if you remember from when we were talking about the cell cycle, remember how we talked about in prophase, the DNA coils up to make chromosomes? Well, for example, in humans, a complete set of chromosomes would be 46 chromosomes. Meiosis ensures that meiosis ensures that you have 46 chromosomes in every cell in your body. Your parents, that's going back a generation, have 46 chromosomes in every cell of their body. Your grandparents have 46 chromosomes in every cell of their body. And if you have children at some future date, they will also have 46 chromosomes in every cell of their body. So one thing meiosis is important about is it produces gametes, it produces sex cells. The other thing, I suppose I ought to put sex cells here. The other thing is it makes sure that every generation has exactly the right number of chromosomes. And there is yet one more important thing about meiosis, because it produces variation. Okay, they may not be big variations, but every single one of us has a slightly different version of the 46 chromosomes that we've got. If you have siblings with the same mum and dad as you, there may be fewer variations between them and you than there would be between, say, you and a person that lives in, I don't know, Australia that you are not related to. But meiosis is responsible for producing variations, small differences that make us all unique, right? It's responsible for making sure we have the right number of chromosomes in every generation, and obviously it's responsible for making sure that we have gametes at all, sex cells at all. So this is kind of what we're going to be looking at. We're going to start here with meiosis, and once we understand how we make gametes, then we're going to look at what happens at fertilization in terms of what happens to your chromosomes. Then we're going to look at what happens when it goes wrong, and we're going to talk about something called karyotypes, because this is, we have 46 chromosomes, 23 of which are in your dad's sperm cells, 23 of which are in your mum's egg cells. That's how we get to 46. 23 and 23 is 46. Half of your information comes from your mum, half your genetic information comes from your dad. Well, karyotypes is what they do when they look at all the chromosomes and they match them up. Because there's like chromosome 1A is an egg cell, chromosome 1B is a sperm cell, chromosome 2A, 2B, 3A, 3B. So you can match them all up. And this explains things how we can determine what your gender is, whether you're a male or a female. It can determine things like sometimes it goes wrong. Sometimes meiosis gets messed up and you end up, instead of pairs of chromosomes, you may have 3 chromosomes. You may have an extra chromosome. And then we get what are called chromosomal mutations. They're not mutations in the changes in the bases of the DNA, but they're mutations in terms of their incorrect pairing of chromosomes. And that's what leads to things like Down syndrome. So this is kind of an interesting topic. And once we've got meiosis all sorted out, the next thing we're going to have is we're going to look at Punnett squares and work out, what your babies are all going to look like. So that's the start of this unit.